Blood transfusions are a cornerstone of modern medicine. They are vital for saving lives and improving health outcomes in countless medical scenarios. They are essential for treating a wide range of conditions, from traumatic injuries and surgeries to chronic illnesses. Without a steady supply of blood, many medical procedures would be impossible to perform safely. However, the world is facing a critical shortage of this life-saving resource. Blood banks around the globe are struggling to keep up with the ever-increasing demand. Every year, millions of people worldwide require blood transfusions, but the demand for blood consistently outstrips the supply. This gap between supply and demand is a growing concern for healthcare providers. This shortage has dire consequences, leading to postponed surgeries, delayed treatments, and tragically, even loss of life. The impact is felt not just by patients, but also by their families and communities. This crisis is particularly acute in low and middle income countries, where healthcare systems are already strained. In these regions, the lack of blood can mean the difference between life and death on a daily basis. Limited infrastructure, inadequate blood donation rates, and logistical challenges in collecting, storing, and transporting blood further exacerbate the problem. These issues create a complex web of obstacles that are difficult to overcome. The World Health Organization estimates that millions of people die annually due to the lack of access to safe blood transfusions. This staggering statistic highlights the severity of the issue. This stark reality underscores the urgent need for innovative solutions to address this global health challenge. Researchers and healthcare professionals are tirelessly working to find ways to mitigate the shortage. The consequences of blood shortages are far-reaching. They affect not only emergency care but also routine medical treatments and surgeries. Patients in need of regular transfusions, such as those with sickle cell anemia or thalassemia, face uncertainty and fear. Their lives depend on the availability of blood, making the shortage a constant source of anxiety. Trauma victims and women experiencing childbirth complications are also disproportionately affected by the shortage, as timely access to blood transfusions can be the difference between life and death. In emergencies, every second counts. The global blood shortage is a complex problem with no easy solutions. It requires a multifaceted approach involving policy changes, increased awareness, and technological advancements. However, amidst this crisis, a ray of hope emerges from the world of scientific innovation: artificial blood. This groundbreaking technology could potentially alleviate the pressure on blood banks and ensure a more stable supply. This groundbreaking technology holds the potential to revolutionize healthcare and save countless lives. With continued research and development, artificial blood could become a viable solution to one of the most pressing issues in global health today. The term artificial blood encompasses two main categories: lab-grown blood and synthetic blood. Both offer distinct advantages and are being actively researched as potential solutions to the global blood shortage. Lab-grown blood, as the name suggests, involves creating red blood cells in a laboratory setting. Scientists utilize hematopoietic stem cells, which are found in bone marrow and have the remarkable ability to develop into all types of blood cells. These stem cells are carefully cultivated and nurtured in a controlled environment, where they are coaxed into producing mature, functional red blood cells. Synthetic blood, on the other hand, takes a different approach. Instead of replicating human blood cells, scientists have developed cell-free substitutes designed to mimic the oxygen-carrying capacity of red blood cells. These synthetic alternatives are typically based on chemically modified hemoglobin or perfluorocarbons, compounds capable of binding and transporting oxygen throughout the body. Both lab-grown and synthetic blood offer unique advantages in addressing the blood shortage crisis. Lab-grown blood holds the promise of providing a near-identical replacement for donated blood, potentially eliminating concerns about compatibility and disease transmission. Synthetic blood, with its extended shelf life, ease of storage, and universal compatibility, could be a game changer in emergency situations and remote areas where access to traditional blood transfusions is limited. The creation of lab-grown blood represents a remarkable feat of bioengineering, pushing the boundaries of what we thought possible in medical science. This innovative process harnesses the intricate mechanisms of human biology to produce life-saving cells outside the body, offering new hope for patients with rare blood types or those in need of frequent transfusions. The process begins with the extraction of hematopoietic stem cells, which are typically sourced from bone marrow or umbilical cord blood. These stem cells are incredibly versatile and have the potential to develop into any type of blood cell. These stem cells serve as the foundation for generating all types of blood cells, including red blood cells, white blood cells, and platelets, each playing a crucial role in maintaining our health. Once extracted, the stem cells are carefully placed in a meticulously controlled laboratory environment. This step is critical to ensure the cells remain viable and capable of differentiating into the desired blood cell types. This artificial niche mimics the conditions within the bone marrow, providing the necessary nutrients, growth factors, and signaling chain more or delicate to support the stem cell survival and proliferation. The environment must be precisely regulated to mimic the natural conditions as closely as possible. Over time, the stem cells undergo a remarkable transformation, differentiating into various blood cell lineages, including red blood cells, which are essential for carrying oxygen throughout the body. The process of coaxing stem cells to produce red blood cells requires a delicate balance of factors, including the right combination of nutrients and growth signals. Scientists have identified specific growth factors and signaling pathways that promote red blood cell development. These discoveries are crucial for optimizing the production process. By carefully manipulating these factors, they can guide the stem cell's differentiation towards the desired cell type, ensuring a consistent and reliable supply of lab-grown blood. One of the key challenges in lab-grown blood production is achieving a sufficient scale to meet clinical demands. This requires not only scientific expertise, but also advanced technological solutions. Scientists are actively researching methods to enhance the efficiency of stem cell expansion and red blood cell production. This involves a combination of biological research and engineering innovation. This includes exploring novel bioreactor designs and optimizing culture conditions to maximize cell yield. The ultimate goal is to create a sustainable and scalable source of lab-grown blood that can be used in medical treatments worldwide. The journey towards making artificial blood a clinical reality has been marked by significant advancements and promising results from clinical trials. These trials are not just a formality. They are the backbone of medical innovation, providing critical data and insights. These trials are crucial for evaluating the safety and efficacy of artificial blood products in humans, ensuring that they meet the rigorous standards required for medical use, and paving the way for regulatory approval. Without these stringent evaluations, new medical products would never reach the patients who need them most. In 2022, a groundbreaking clinical trial took place in the United Kingdom, marking the first time lab-grown red blood cells were transfused into human volunteers. This was a monumental step forward in the field of artificial blood research. The trial aimed to assess the safety, and longevity of lab-grown blood compared to standard blood transfusions. Initial findings were encouraging, demonstrating that the lab-grown red blood cells function similarly to donated blood cells, which is a significant milestone, and remained in circulation within the recipient's body. This longevity is crucial for the practical use of artificial blood in medical treatments. Another notable trial conducted in Japan in 2022 focused on testing hemoglobin vesicles, a type of artificial oxygen carrier in healthy volunteers. This trial was essential for understanding how these vesicles perform in real-world conditions. While the trial demonstrated the potential of these synthetic substitutes to transport oxygen effectively, some participants experienced mild side effects such as fever and rash. These side effects, while minor, highlight the need for further refinement. These findings highlight the importance of ongoing research to optimize artificial blood products, making them safer and more effective for future use, and minimize potential adverse reactions. Continuous improvement is key to the success of any medical innovation. Despite these promising developments, it is essential to emphasize that both lab-grown and synthetic blood are still in the research and development phase. Extensive further research and larger-scale clinical trials are necessary to fully evaluate their long-term safety, efficacy, and suitability for widespread clinical use. Only through rigorous testing can we ensure these products are ready for the medical field. Scientists are diligently working to address remaining challenges, such as improving the stability and storage of artificial blood, such as optimizing production methods, ensuring product consistency, and scaling up production to meet potential future demand, and conducting comprehensive safety assessments. The future of artificial blood is promising, but it requires meticulous and sustained effort to become a reality. Thanks for watching. If you found this informative, please like and subscribe for subsequent videos, as well as to support our channel.